into changes in the lifespan and in the neurodegeneration of these organisms. Okay, having introduced myself, I'll leave you now my email here. So if you have any questions later on, or if you want to contact me, you have the email. We also have an account for the lab, both in Instagram and in Twitter. So I'll be happy if you join us in these social networks. You can see the kind of things that we do in the lab, and we make a lot of sort of outreach and talk about the projects that we do and put lots of pictures and all that. So we can start now, and I will have to tell you that I like these classes to be sort of based on a kind of dialogue, which means that I am very welcoming to questions. And there's two ways that you can do these questions. One is by using the chat that we have here on the right, and you can ask questions anytime. I have an alarm, so I will hear anytime that somebody puts a question up. And you can also use the hand here at the bottom where you have a little doll with the hand up. So if you click that, then I can give, if you want to use the microphone, I can then give you permission to use the microphone. So, okay, so let's start. I know now that the majority of you thinks that we cannot live forever. And so we will start from that. And we'll start a little bit about talking about aging as a sort of general concepts of aging. Okay, so we're now here. And so this is an excerpt of a book by Michel de Montaigne that he wrote at the end of the 16th century. And basically it says something which is very important to start this class. It's a sort of kickoff for the class. And so it says that to die of old age is a death rare, extraordinary, and singular, and therefore so much less natural than the others. So here you have to be aware that this was written in the 16th century, okay? And we will comment that on in a second. It is the last and extremest sort of dying, and the more remote, the less to be hoped for. It is indeed the boundary of life beyond which we are not to pass, which the law of nature has pitched for a limit not to be exceeded. And so if we were going to give the poll to Michel de Montaigne, the poll that I just gave you, he would definitely say no, because he defended that there is a limit, there's a border, which is the limit of life, which we cannot pass. And in the 16th century, most of the people were dying not of this old age that it mentions in the first line here, but they were dying mainly of infectious disease. And so to die of old age was a death rare. Nowadays, it's not anymore. And so we're lucky enough that we have, in the 21st century, eradicated many of these diseases. And I don't know if you are listening to me well or not, but I will remove the camera so then we don't have problems in the future, okay? Now that you have seen me, I will close it. Okay, so you have this view that there is a border that we cannot pass. And aging has been in the minds of humans and the minds of mankind for many, since the beginning of culture and the beginning of everything. And so we have very good expressions in culture that reveal this interest and this fear of getting old. So from here on the left, we have a painting from the 15th century that shows the two big polarized phases of life, being old and being very young. And then two literature works, one from Oscar Wilde, which is I guess most of you will know, The Picture of Dorian Gray, which is the story of a man that does not get old. And so he has this special way of maintaining himself always young, with the consequences that might have. And then on the right here, a book by Scott Fitzgerald that became very famous because it gave rise to a movie in 2008 already, which is this one called The Curious Case of Benjamin Button. Benjamin Button was somebody that lived the life the other way around, which meant that he was born old and then would regress in age as life went by. And so this wonder about what happens in later in life and what makes us old has always been there, and it's always been behind our minds. And nowadays we have the tools and we have the knowledge to tackle many of the questions of why we get old, and if there's any way that we can extend this lifespan, and we can extend what is now called the health span. And I will go into that also in a minute. What we know now is that even though Michel de Montaigne said that die of old age was a death rare, we know that now dying of old age is not a death rare. And if we compare these two maps here, it's very clear to us that from 2015 to 2050, the percentage of world population over 60 years old is going to increase immensely. So whereas Here we see that in the developed world, the percentages are here, and so the reddest is the highest and the green is the lowest. We can see that you have, old population in Europe, and places there actually is actually quite young. But as the years go by, what we can see here is that most of the world in 2050 will have a population over the age of 60, which will be more than 30%. So this is a huge problem, because this is not just the fact that you get people to get very old, and this is a very good idea because you get people that we are able to extend people's average lifespan, but we have a problem, a social problem, which is what's going to happen to the world with all these people that are very old. And so part of the science and part of the research that is done nowadays is meant to study how to expand this life limit, and also how to keep people very healthy until very late. Okay, so this is my seminar today. It's quite a complicated, it's quite a challenging one, because what we have to do today in 2 hours is to talk about, give you an overview of aging, focused on 2 main things that we can study to study aging. But on the other hand, we're going to leave many things on the side. And so then, of course, this is not a full course on aging, so we will not have everything. You also will not have the discussions of the papers that we usually do when we don't have to be in confinement. When we have these tutorials with all the students, we have then another 4 sessions where we can discuss papers. Like this, you'll have to just take this seminar, and we have to make the most of it. And so how did I divide this? I divided this starting with these general concepts that we started already, then moving to how to study aging using people, not using people as in using people, but as in studying different populations. So the population of the people that are very old and they reach an age of over 100 years old, and they're called supercentenarians, compared with people that have premature aging. And most of these are people that suffer from rare diseases that have, as a consequence, premature aging. Then we will talk about genetics of aging and ways of tackling the study of aging. How do we study aging? How can we study aging in a lab? And then at the end, I'll give you a sort of very brief and broad overview of the hallmarks of aging. So all the things that are being studied now in order to know more about aging. Okay, so you're welcome to interrupt me whenever you can, whenever you want, and we can then have a conversation. I will also give you a little bit more of these polling sessions throughout the talk. Okay, so if I was then going to ask you now, if we were in a classroom, I could then ask you, what do you think aging is? But most of the times, we kind of all agree that aging is about becoming older. And it's not sort of a linear process, it's a multidimensional process of many things: physical, psychological, and social change. So as we get older, we change, and these are many different sides of our life. But there's a very important thing about aging, and when we talk about aging, we can be talking about mainly 3 different things. One is that aging can refer to cells that have stopped dividing, and this is called cellular senescence, and we have a picture here of some cells that have stopped dividing. We can talk about aging as the process of becoming older, which is most of the times what we think when we think about aging. We think about people becoming older. And this is organismal aging, and we have the picture of the old man from the painting. But then we can also talk about the aging of a population, and that is population aging. So we have the population data, we see maps, and we see where are the places in the world where people can live the longest, where are the places, other places in the world people don't live as much. And so this is population aging. So the 3 things can be studied in different ways, and the 3 things can give us lots of clues in order to study aging and the genetics of aging. So we can refer to aging also as senescence, not just to cells, but we can talk about organismal senescence. So senescence, or the process of aging, is achieved in cells. Cellular senescence is achieved after a defined number of cell divisions. And towards the second half of this seminar, I will tell you more about this cellular senescence. But we also can talk about organismal senescence, and this is characterized by the declining ability to respond to many things: to stress, to the increasing homeostatic imbalance, to the increased risk of disease, which are all things that happen as we get older. And these are the signs of aging. So if I was going to ask you now, what are the signs of aging? Many of you would say, okay, well, you get people that the skin gets older, so you get more wrinkles. This is one sign of aging in an organism like humans. our skin gets more wrinkled as we age. There are other things like the increased risk of disease, the increased risk of cancer. All of these are signs of aging. And then you have two very important concepts, which are these: average lifespan and maximum lifespan. And now we will do a small poll, and I will. One do you think has changed the most? So let's say, which has changed the most in the last century? Or so now you have a poll, and so I want your opinions about what has changed the most in the last century: the average lifespan of individuals or the maximum lifespan of individuals? Okay, so we're getting into the end. I will show you the results in a minute. And there is a large majority of people that have voted that the thing that has changed the most in the last century has been the average lifespan of the population. And if I had asked you this about what has changed the most during all the lifeline of humans, the answer will be the same. And whereas in the first question I asked you, there was not really right or wrong answer, in this case there is. And in fact, I will show you now that the most of you have had. So average lifespan is what has changed the most in the last century. And in fact, if we want to be very blunt about this, if we look at the maximum lifespan of individuals, that has not changed the slightest in throughout all the human life on Earth. It's very difficult to say this because obviously only in the last 80 years, maybe, we have enough registry in order to actually say for sure that people have a certain age. There's always been claims of people that said, oh, I've been living for 350 years. There's always a man in the middle of the mountains that says that has lived for 175 years. But those things cannot be proven, and therefore they're not used as data. And so what happens is that we know, and I'll go back to the presentation, we know that average lifespan is what has been changing the most especially over the last 100 years, whereas maximum lifespan has always been there. If you were lucky enough that you would not die of an infectious disease that affected most of the children in the world, and then you would not die of an accident at the age of 25, there were people in the population that always achieved 100 years old. The difference is that these people that achieved the maximum lifespan, they were always much less in numbers than the ones that can achieve maximum lifespan nowadays. So it's a matter of numbers, and which is why average lifespan is the one that is changing. Average lifespan is the same as life expectancy. When we talk about the population, it's a statistical measure of how long an organism may live in a certain environment. So we say, okay, in Spain is one of the countries, sometimes is the top and sometimes is the second, where people live the longest. Why? Because average lifespan in Spain is very high. Maximum lifespan, it doesn't matter where you live. The difference is that in places where not as advanced as in others, less people will achieve this maximum lifespan, but there's going to be people that achieve the maximum lifespan. Okay, so this is the age at which the oldest known member of the species has died. And so far, what we know is that the oldest member of the species, of the human species, has died at the age of 122, which is why that many times we comment, can life be expanded beyond this 120-year limit? Why? Because there are a few people that age, but then there's no more, and there's no record of anyone ever living more than that. So we can get into very many philosophical discussions about this. Okay, so mean lifespan varies with susceptibility to disease, accident, etc., whereas maximum lifespan does not. There's always 1 or 2 cases, or 5 or 50, that will achieve that maximum lifespan. And this is just a table. It's from Italy. It could be from Spain. It's the evolution of life expectancy or lifespan or average lifespan in Italy from 1861. So if we look at 1861, the average lifespan was 29 years old, which meant that most of you would now be at the end of your life. But only in 150 years, we have now increased that average lifespan to 82. 82 years old, the average of the population. Obviously, always in general, women live longer than men. And so we've gone from 29 to 82 in a mere 150 years. And this is a huge advance. And it's all due to the medical attention that people have now, to vaccines, etc., etc. If we look at graphs, and this is not a real scientific graph, but it's just an example, we have the percent of survival here, you have the age of the population here, and as you can see, the difference that there's always a person arriving to be 100 years old, but there's a lot less arriving to be 100 years old 50,000 years ago than there were in 1970. But the main difference is in that part of the curve here, which is the average lifespan. So we can be very happy now because we have much higher average lifespan than the majority, well, than all the humans that lived before us. And this here is an example of maximum lifespan and an example of the maximum lifespan that we would like to achieve. On the left, so I forgot to tell you, I am a professor at the University of Barcelona. I do research here, but actually originally I am from Portugal. I spent a lot of time in the UK, and then I came to Barcelona. And so Manuel Oliveira, this guy here on the left, is a film director from Portugal. And he died in 2015, aged 6. And this is Manuel Oliveira in 2008, when he was 99 years old. And as you can see, he started dancing very slowly with these people that are in this group of performers from a theater. There's music and everything, but I didn't put it on. And what will happen now is that he will gradually just get rid of his stick and continue dancing, and at the end you will see him running. And so this is what we want. We want to achieve maximum lifespan, but we want to achieve maximum lifespan doing this. The majority of people that get to be 99 years old, they're not as healthy as Manuel Oliveira. The same way they're not as healthy as this person here on the right, P4RTE Singh, that in 2011 was 100 years old and was still running marathons. Slower than many other people, but he was still running marathons. And so what do we want? Do we want to extend beyond 120, or do we want to be healthy and then die at 120? There's not a yes or no or situation. Of course, if we can, we want both. But the focus now here is, in fact, of getting people, not just the quantity of years, but the quality of the years. And so we want to get people that are living longer, but they're healthy. And so we start talking not also of health span. Okay, so here on the left, what we have is the age of the old. So by 2050, like the map that we saw, we will have, and it is already growing exponentially, the number of people over 60 is going to be much higher than what we have now, which we're about here. Okay? This is a problem that we have, and so the more healthy we can keep the people that are getting older, the better. And so we want people to be old and not look a day over 50 years old. Okay, so can we extend this life beyond the 120-year limit? Well, we don't know, but the research is focusing not just in extending lifespan, and that's what we do using model organisms, and we will get into that, but we also want to keep this, not just these organisms living for longer, but we want to keep them living for longer in a healthier state. So research has to focus not just on the increasing lifespan, but also in increasing this health span. We don't want to eradicate aging, but we want to keep people healthier for longer. And this is what we have here in this graph. At the moment, what we have more or less is this kind of curve. We have people having the maximum quality of life until they are the age of 20, and then that starts gradually coming down, as you can see here. So for you, you are all more or less in the maximum quality of life. I cannot say the same about me because it's already going down. And then as this goes, and lower quality of life. And so many times this actually goes a bit more like this. So what we want is not this gradual decline in quality of life. We want a plateau where the quality of life is always good, and then like a decay which is fast and at the maximum lifespan. So we don't want the prolongation of suffering, but the prolongation of normal vital function and quality of life. Okay, so enough of all these general concepts. Is that we think about aging as a way of maintaining people living longer, but healthier state. And we will be able to do that by studying aging, knowing more about the molecular biology of aging, and knowing more about how cells behave and how we can actually modulate these cells so we can keep people healthier for a longer time. Okay, so how can we study aging? There's many different ways of studying aging, and we already saw there's 3 different ways of looking at aging: cells, individual cells, people, individual people, and whole populations of individuals. So we can look at these 3 and take conclusions and make hypotheses, studying all 3 of them separately, or trying to make the most of studying them all 3 together. Of course, there's different scientists studying each of these. And one of the ways that we can actually study aging is by doing experiments. So this is the oldest way of studying aging, is by doing experiments on ourselves. And this is what happened to this person here. This man here, it was called Luigi Cornaro, and he was an Italian, very, very wealthy man that lived in the 15th century. He was very wealthy, which meant that he had lots of money. And so he was very wealthy, but he was not very healthy because he had lots of money. So he could have a life of partying and of excesses where he was doing all sorts of different things, eating too much, drinking too much. And he was very ill at the age of around 35. And so he had read many things, and he came up to the conclusion that he had to start doing dietary restrictions in order to increase his health span and to increase his lifespan. And so he started doing experiments on himself, reducing the amount of calories that he would take every day. And so gradually he started recovering his health. And so by the end of it, he had the secret to living up to 100 years old, which is this one here. He was ingesting only 350 grams of food per day and 440 milliliters of wine. So wine was not ever out of the equation. With only this amount of calories, he managed actually to live even though he was very ill and nearly dying of gout and obesity and all of that at the age of 30 something, 40, he was able to live until the age of, well, we don't know quite certain that if it was between 98 and 102, so around 100 years old. And so he was very famous, and he wrote a book called Discourses on the Sober Life. Even though he was ingesting 414 milliliters of wine every day, he wrote this book, which was written in Italian, and it became so famous that it was actually translated in many different languages. The second title of the book was How to Live to 100 Years. And so since at least the documented first experiment of extending lifespan of Luigi Cornaro, we know that dietary restrictions are actually very much correlated with health span and with the increase of lifespan. Okay, but it's not very good to start doing experiments on ourselves, and nowadays we don't have to do that. So we can study other things. And here's a list of the keys to this biology of aging. We can study the longest living humans. So we can now go and study the genetics of people that live beyond 100 years old. These are called supercentenarians, as we were saying before, because they get beyond 100 years old. They can be 100, 105, 110. So we can study these people. On the other side, we can also study early aging syndromes. We can study people that suffer from diseases like progeria, that are diseases that increase the rate of aging. So they will age faster than the rest of the population. They will be people that get an older person much earlier. From these studies, we can then, look at the genetics and see if there are any aging genes. Then we can look and build biological models to study lifespan. This is where we get model organisms into the equation. And then we can study molecular and cellular mechanisms related to all of the things that we had studied before. Okay, let's start with these longest lived humans. So we have the 2 documented nowadays. These are the 2 people that have lived the longest. Jean Calment, that lived to 122, that was born in 1875 and died in 1997. And Mr. Kimura, from Japan, that lived to 160. In between now, there are many people that have beyond 100 years old and have 110, 115, 112. But nobody has been reported to be older than 122. And no male has been reported to live beyond 116. Okay, so these are the sort of 2 top. Then we have Jeanne Calment. Jeanne Calment has a very funny story. She was very famous while she was still alive, and apparently she used to give interviews telling the press that she stopped smoking at the age of 100. And so she was this sort of icon in France of you can actually have fun and still live to 122. What can we learn from these people? Well, we can learn that genetics is important, but also that is a very, very strong influence on aging, which is from the environment. And so there's all the inputs from life, not just from the toxic and chemical drugs or food, but also from psychological states, so many different social interactions. So there's many population studies being done nowadays that study different environments where are different regions in the world where people are documented to live for longer. And these are the people that maybe have a better genetics, but they also usually have better environments. What do we know from this? Well, we know from twin studies, and in twin studies, we know that from twin studies we can learn a lot, because if we have twins that have genetically identical and they live in different environments, we can study the influence of the environment in the genetics. And from twin studies, it suggests that genetics accounts for only 20 to 30% of the chance of surviving to be 85. So that's quite a low percentage. The rest are lifestyle choices, particularly diet, exercise, smoking and drinking, of course, like Luigi Cornaro. So if you were not drinking and eating too much, you would not have been nearly dying at the age of 40. And these habits play a more important role in lifespan than, in fact, genetics. However, we know that becoming a supercentenarian can run in families. So there is a genetic link. For instance, in the family of Michelle, no, what was her name? Of Jeanne Calment, the family of Jeanne Calment, there are many people that live very long, and there are many families that are associated with people that live very long. And so supercentenarians are good genetic models to study aging. Okay, so what can we do? We can do the whole genome sequences of the world's oldest people. We can go and look at the world's oldest people, sequence their genomes, and take conclusions. And this is only one of them. There's many, many different studies like this. This was published in 2014. And what it says here is a very important sentence. It says, we found no significant evidence of enrichment for a single rare protein altering variant or for a gene harboring different rare protein altering variants in supercentenarians. This means that there is, in most of the studies, we cannot find just one variant which is involved in the genes controlling lifespan. We have to take into account many different things. And so it runs in families because families have the same genetic background. But when we start pinpointing different variants, we cannot come up with the real conclusion of all these variants is all you need to get to live for over 100 years old. Okay, so then we can study the second part, which is supercentenarian versus progerian, okay? What is supercentenarian and what is progerian? You are very quiet. Are you all okay? Nobody wants to ask me any questions? Okay, so I'll carry on. So here we have one of the families that also has been documented to have people live very long, the family of the royal family of the United Kingdom. Here is the Queen. Her mother died over the age of 100. She was probably going to also last for longer than 100 years old. But so we can study the royal family of the United Kingdom. But we can always, okay, there's a question. I'll go back to here. Was there alternative medicine as a factor for living longer? What do you mean, as in the environment? I'm not sure if I understand. I thought in the slide that I have it. Okay, let me go back. Probably because there's lots of connections and there's lots of studies. See, yes, because there is also, okay, don't quote me on this, but there is a study that has correlated faith to longer living people. And so there's a Bernard, this is for you. There's a population of people that live in California that live in a kind of sort of commune where they are living in a very small community, and they are very old. They are very old, and they actually have in the United States, one of the longest average lifespan of the rest of the population. And so this is called Loma Linda or something like that. And it was very documented because of that. And so it was studied by all the people that there are many factors that contribute to the fact that these people live longer. One is community spirit. And this is not the joke, because of course, if you live in a community and there's community spirit and people help each other, it's a lot easier that they're going to live longer when they're older. But there was also a connection with faith. So the people that were religious lived longer. Of course, all of these correlations have to be taken very, very, very carefully. And so maybe I should delete that alternative medicine from that slide. But it is true that if we start looking at people that living in a community and perhaps they all have faith in a specific religion, perhaps we can make a correlation. But correlations are very dangerous. Okay, so we have to use them with a pinch of salt. Okay, so the Queen of England. Many people also said that the Queen Mother lived very long because she drank lots of gin and tonics, and so we're not going to believe that as a correlation. Just because she had lots of gin and tonics, that does not necessarily mean that this is why she lived over the age of 100. Okay, so what do we have? We have the super centenarians, and we have the ones that are young, but they on the other side of the coin, we have the ones that are young, but they have older appearances, and they have what we call the progeroid syndrome. And these are very much documented, and most of them are people that have rare diseases. These are called progeroid syndromes, and within the progeroid syndromes, there are many different ones. The most famous one is called progeria. It's actually called Hutchinson-Gilford progeria. And then we have Werner syndrome, Cockayne syndrome, Bloom syndrome, lots of different syndromes, that what happens to people is what is here in the photo. This is Sam Burns. He was 17 when this picture was taken. And he looks, so he had outside sides of aging of a much older person. And it's not just the outside sides of aging that made him look like an older person. He had many different pathologies associated with age. So he had atherosclerosis, he had heart problems, he had muscle problems. All of these is associated with an older age. And this is not just Sam Burns, but all the people that suffer from this progeria. And then on this side, we have Jean Baptiste, which is a child that suffered from Cockayne syndrome. Cockayne syndrome is also one of these progeroid syndromes. And so what happens to children, even though they have some neurodevelopmental abnormalities and some other developmental abnormalities, that when they are born, they're usually more or less okay, but then they start aging very fast. And so you can see here by the age of 7 what they look like. And also, this is it's a very interesting disease, because not just they look older, but they also have very high incidence of neurodegeneration. So these people that suffer from Cockayne syndrome have higher neurodegeneration than the people that don't suffer from Cockayne syndrome, of course, and they have faster the neurodegeneration, which is associated many times with aging. Okay, so these progeroid syndromes, there's a long list, longer than what we have here. Most of them, apart from Hutchinson-Gilford progeria, all of these are defects. So because they're rare diseases, most of them are mutations in genes that are implicated in DNA repair. So you have Werner syndrome, Cockayne syndrome, xeroderma pigmentosum, trichothiodystrophy, and many others, which are genetic mutations that affect proteins involved in DNA repair. In the case of Hutchinson-Gilford progeria, what we have is mutations that affect lamin A or C, and this affects nuclear architecture. So people with Hutchinson-Gilford progeria have problems in nuclear architecture, and that is thought to affect genetics of these individuals. We're not going to get into all the biochemical part of this, but we need to know that these progeroid syndromes are a way of knowing genetically what is happening during aging, because if you can have a single mutation in a gene which is implied in DNA repair, and this person is going to age faster than the rest of the population, this is already telling us something about the implication of these proteins in the normal process of aging. And if we can study the supercentenaries and look at variants and see what the different variants do, then we can also take conclusions about what might be implicated in the fact that these people can live longer than the majority of people. So we can study the genetics of aging. So, okay, let's start with this DNA repair Cockayne syndrome people. This is the progression of one of these children. These people have recessive mutations in 2 proteins, CSA and CSP. Both mutations give rise to a syndrome called Cockayne syndrome. And these people, what they have is a defect in a pathway that repairs damage from the DNA. And I don't want to get into the details. this is only one of the pathways that is involved in DNA repair. There's many different others, but I have another question. How many years? I think some, well, they don't get to live much longer than 20. Sam Burns, the one that is in the picture, I think he died. He was very famous because he started the foundation, and so. But I think they die around the age of 20. Most of them die in their teenage years. Cocaine syndrome patients, most of them will die early teens, so in about 12, 13 years old. So it's quite a serious one. Then there are other progeroid syndromes where people, like Bloom syndrome, there was a picture of a Japanese woman, where people can live longer, but they will age faster than the rest of the population as well. Okay. Okay, so we have cocaine syndrome. We have two different ways of repairing DNA, and there are many different other pathways. So I just wanted to focus that what we have here is damage. So you have damage to your DNA, and then there are complexes of protein that in this case is called nucleotide excision repair that are able to go into the DNA, recognize the damage, excise the damage, and then make new DNA which is not damaged. So this means that if we have this damage and it's repaired, you get back to having a normal DNA chain. If you have damage and it's not repaired, two things might happen. Either you keep the damage, but if you're a cell that is dividing, then you will propagate this damage as a mutation. And so we have two different ways of looking at this. We have to look at the way to look at DNA damage as mutations, and the way to look at DNA damage as only DNA damage. There's a problem with the sound. Okay. Can you hear me okay now? Hold on. I will switch off and I'll switch it on again. Okay. I switched the microphone off and I switched it on again. And you're hearing me okay. Okay. In this morning, we had a slight problem like this, but we thought it was the camera. So I removed the camera, but now I have no camera. So we will see. Okay. So now is the moment where I'm going to ask you to make another poll, since we're making it anyway. So I will just poll you again. and so I want to ask you a yes and no question, which is, the question is, are the majority of the cells in our bodies dividing? So as we live, dividing. Are the majority of the cells in our bodies dividing? This means, start new, I still have the other one on. So are the majority of the cells in our bodies dividing, meaning that the majority of the cells that we have now, okay, okay, I'm happy that it's fine now. I didn't do anything. Sometimes internet goes up and down, and we have lots of people in class, so that could be one of the problems. Okay, so there's a poll now here that is asking you if you think the majority of the cells in your bodies are dividing now, as we are speaking, during your daily life. What is happening to the cells in your bodies? You cannot answer. I don't know why. Why? Okay, I will do something here. Okay, now try again. Okay, now it was a problem with showing responses. Okay, it's very exciting to see the results, because we're a moment where we have the same votes for yes and the same votes for no. Okay, the 10 people that are left, please answer and see what happens. Okay, maybe these 6 are not here, and so we will see the results in a minute. Okay. Okay, I'll show the results now, and so 23 people answered no, and 23 people answered yes. So this means that in fact, these people that answered no, they are right, because the majority of the cells in our bodies are not dividing at the moment. So think about something which is usually quite a good example. Think about Chemotherapy. People that unfortunately have to go through chemotherapy because they have certain form of cancer, they will go through different rounds of chemotherapy. Do you know how chemotherapy works? Chemotherapy is a carcinogenic, so it's a mutagenizing agent that what it does is damage the DNA. And so it does damage the DNA. There's a certain number of checkpoints. This is not part of this class, but what will happen is that chemotherapy will make the cells of the body stop dividing. Okay, as in a general way. I mean, we could get into details about this, but the idea is that agents like therapeutic agents, what they will do is they will induce death in the cells that are dividing. Okay, and so what happens to people where they have going through treatments of chemotherapy? They have gut problems because the gut cells are the ones that are dividing quite a lot. They lose their hair. They have problems in all the renewable tissues in their bodies, but most of the cells in our bodies are actually not dividing. Okay, because most of the cells in our organs are not dividing. So unless we are a developing embryo or we are a growing individual in sort of during childhood. Okay, so having done that, we can now talk about damage. And okay, so let's go here. So we have two different types of ways of talking about DNA damage. So one is that then there were two theories about this. The first theory stated that somatic mutation theory of aging. And so the somatic mutation theory of aging that Gioacchino Faglia stated in 1958 said that the accumulation of mutations in the genetic material of somatic cells with age results in the decrease of cellular function. Fine, there's nothing wrong about this. But then a few years later, there was another theory, which is the DNA damage theory of aging. And this one says the aging is a consequence of unrepaired accumulation of naturally occurring DNA damages. And so here is where we have to make the big difference between a dividing cell and a non-dividing cell. If we have DNA damage in a cell which is not dividing, this damage can be repaired or not. If it's not repaired, it stays in the cell. No mutation appears because the cell is not dividing. Whereas if you have a dividing cell and you have damage and this damage is not repaired, what will happen is that it can give rise to mutations because the cell is dividing. It will only give rise to mutations here if the cell is dividing. Otherwise, damage is not going to give rise to mutations because the DNA is not replicating. Therefore, there's no propagation of mutations. What is DNA damage? DNA damage is what happens to our DNA all the time. And so this is when you get all depressed, because this is the frequency of occurring DNA damage in your cells. 10,000 to 500,000 molecular lesions per cell per day, which means that we are constantly being damaged by different sources. Some of them are endogenous, like reactive oxygen species, errors of recombination, errors of replication, things that are a result of the cellular metabolism that may damage our DNA, and exogenous agents, which are the ones that we are most used to. So UV light, if we're all aware of the associations between going to the beach, sunbathing, and skin cancer. This is UV light damage. Then you have the DNA itself could activate oncogenes, thus activate division in breast. But for that, the DNA, okay. So the DNA damage itself could activate oncogenes. Why do you say that? Let's see if I understand your question, because are you trying to say that if you have damage in the DNA, this can activate an oncogene? Is this what you're trying to ask? No. So if you have damage in the DNA, what will happen, for instance, here, let me go back. If you have damage here in the DNA, you have damage. If nothing is happening, this is global genome repair. So you have your genome and there's some damage, but nothing here is happening. And so if nothing is happening here, nothing is going to happen. So you have damage and it stays there. What happens is that you might have a problem then when this cell divides. If the cell divides, this can become, this place where the damage is can become a mutation because has not been repaired, and therefore can become, if it changes the basis of the DNA, for instance, it can become a mutation. So you generate a mutated DNA, and this propagates in the next generations of cells. If you don't have replication, you have a cell here, and there's transcription. Transcription goes through in the same DNA. The transcription finds a place with damage, and what might happen, and most of the times what happens is that this machinery is not able to then transcribe the DNA. And so then it stalls. So you might have problems only at this point, which is In this case that you're saying that DNA damage can affect an oncogene, only very slightly if you have just here the transcription of something which is involved in an oncogenic process, okay? This is the only way that you can have the damage interfering with something like that, by not being able to transcribe whatever is around it. Did that answer your question? No. Ada, are you still there? I did not convince you, no? Okay, we will continue. If you have then more questions, we can discuss this later. But the idea is that when you have damage, unless you have a mutation, you're not going to mutate the protein. So you're not going to produce a mutated protein. That's the whole point of it. You might change the way proteins are transcribed if you accumulate damage, and then you have differences in transcription. You might change the epigenetics of these cells, okay? But you're not going to create mutations in the genes and then create proteins that are mutated, okay? Where was I? Okay, here. So DNA damage versus mutation. In contrast to DNA damage, a mutation is a permanent change in the base sequence of the DNA. So a mutation is then part of the genetics of the cell. If you create a mutated cell, this cell will continue dividing and continue being mutated. If you create an individual which is mutated in its germ cells, it will create progeny which is mutated because the germ cells were mutated. If there's only damage, it might change the homeostasis of the cell, but it's not going to create a mutant cell exactly. That a mutant cell in the sense that this cell can pass the mutation on to generations. Damage can be repaired, whereas a mutation cannot. So you can repair damage, but a mutation, you cannot repair it. A mutation is going to be a change in the basis of the DNA, and therefore is going to be treated as normal DNA, and therefore it will be continued passing on generation to generation of cells or of individuals. Mutations are replicated when the cell replicates, so this is what I was saying, it will pass on, as the cell divides, it will pass on this mutation. And then repair DNA damage can induce mutation. In dividing cells can induce these mutations. What will happen then? So if you have non-dividing or slowing dividing cells, you have to have a big difference between the non-dividing or slowly dividing cells with the cycling or proliferating cells that are dividing a lot, between cells that are not dividing. In non-dividing cells, unrepaired damage accumulates over time. Okay, so you have non-dividing cell, and if the damage is not repaired, as we were saying before, it will just accumulate over time, because nothing is going to repair it. In dividing cells, this unrepaired DNA that do not kill the cell by blocking replication, or do not kill the cell because there's a checkpoint and it will decide this cell to go through apoptosis, will tend to cause replication errors and then cause mutations. And so in non-dividing cells, we have mutations. In dividing cells, we can generate mutations. In non-dividing cells, we have accumulation of DNA damage. And so the theory mainly says that DNA damage in dividing cells may give rise to cancer or other types of mutations in clones of cells, but DNA damage in non-dividing cells is likely to be a cause of aging. And so this is damage accumulation in cells over time, cells that are not dividing. As we were seeing before, the majority of cells in our body. Okay, is that clear? I went too fast now. Yeah, okay. So the question is, can damage generated in one individual be transmitted to the progeny? The only way that you can transmit to the progeny is, in fact, by creating a mutation. Why? Because when you're talking about transmitting it to the progeny, it has to be in the germ cells. If it is in the germ cells, one of the ways that germ cells are dividing, and they go through meiosis, so not just the female ones, but and the male ones as well, and that will create a mutation because the cells are dividing. Well, as you are doing meiosis, you will create mutation. So that damage can be passed to the progeny because it's a mutation. If it's just the damage, eventually it's not going to allow the cell to divide. The cell will die, or will then create a mutation, which then be passed to the progeny. Okay. Okay, so this is not something that you really need to know. I just want you to know that there are many ways of damaging DNA that can be endogenous and exogenous, and there's a little list here. This damage can be repaired. There's many different DNA repair mechanisms. So you have base excision repair, you have nucleotide excision repair, you have more than the ones that are listed here. And these deal with different lesions. For instance, nucleotide excision repair, which is the one of Cockayne syndrome, deals only with lesions that have things that attach to the bases of the DNA. There are others, like base excision repair, that are involved in base mismatches. So if during replication a base that should not be inserted in the DNA is inserted, then base excision repair comes and gets rid of it before the cell divides again, etc., etc. You have double strand break repair, which deals only with double strand breaks. Okay. And so what happens is you have two different situations. You have a healthy cell, and in a healthy cell, a rate of DNA damage has to be equal to the rate of DNA repair. Otherwise, you will accumulate damage. In a diseased cell, the rate of DNA damage is higher than the rate of repair, therefore this cell will accumulate damage. And so because there are so many events of DNA damage per day, you have to say that all of the rate of repair has to be very high in the individual all the time. And if this is happening okay, then there is no problem. The cell has damage, but this damage is repaired, and so there is no problem. The problem starts when this damage is not repaired. Then you can have damage in nuclear DNA, you can have damage in mitochondrial DNA. This will lead to pathology, and the pathology can be cancer in the case of dividing cells, or senescence in the case of non-dividing cells. And in the case of dividing cells, you can also lead to cell death. Okay, so now it's 4 o'clock. I will finish the part of DNA repair, and before we go through the next part, we will make small reading. So I think it works quite well, unless you don't want to. And if you don't want to, tell me. I'm not going to do a poll about this. Okay, so DNA repair and aging. Many different DNA repair pathways exist to deal with all kinds of DNA damage. DNA damage accumulates in aging cells. Why? Because DNA damage keeps accumulating in cells. If DNA repair is not working properly, it will accumulate more. But even if DNA repair is working properly, maybe we will accumulate damage that escapes the DNA repair. Failure to repair the DNA can lead to aging, because we also know, and this is a loop, that the mechanisms by which cells repair DNA also, their activity decreases with aging. So on the one hand, we have DNA repair that decreases with aging, because we are aging and the proteins in the cell are also, their activities are lower. And so because of that, more damage gets accumulated, and more damage can accumulate and also lead to more senescence in the cells. Okay, so now we can have a break. I will leave everything on, and it's 4:01. Let's come back at 4:10. You can go and have a walk at home and have a coffee if you want. Okay, we'll be back at